So I'm Dr. Brian Kaufman. I'm a family doctor with CLL, doctor turned patient.
And my name is Dr. Tom Kipps. I'm at the Moores Cancer Center at UC San Diego, and I've always been a lifelong student of this disease, but actually right now we have some very exciting lessons in store for everyone. I think it's an exciting time in CLL and cancer in general as we get away from some of the strategies that have been employed for over 50 years to treat cancer. We're getting actually smart about how we go about treating these diseases.
So we're at ASH 2012, and this is an incredibly exciting time in CLL. So what's got you really excited?
Well, there's a number of things. I mean, one has to really appreciate the fact that there have been a lot of work on the biology and genetics of leukemia and cancers in general. And right now, all that work is bearing fruit because we have a really good understanding of how cancer may differ from normal cells. And we can also pick upon areas where the cancer cell may be more susceptible. And so therefore, we can go in with very specific agents that might be able to harm the cancer cell without harming normal cells in the body. And so this is a paradigm shift. In part, we had the history of chemotherapy, which is really on the idea that cancer cells grow fast, and the reason that we have tumors is because there's uncontrolled cell growth. And so for many years we employed strategies that would really kill off rapidly dividing cells. Unfortunately, we know now that those treatments have some long-term consequences as well as some short-term difficulties in truly achieving a remission that's long-lasting in many cases. I think what's exciting about this year's ASH is that we have the plethora of new drugs, that many of which are orally active, that really go after key signaling points within the cell. I think what has me most excited are a number of these drugs. In particular, there's been just a tremendous improvement in targeting this molecule Bcl-2. Bcl-2 was first identified over 20 years ago or more. It was B-cell leukemia 2. So it was a gene that was identified in a related cancer, follicular lymphoma, and that was the site at which there was a translocation in the genome. And so when you see that, you know that that is playing an important role in the cancer. The real question was, what is it doing? And then after you find out what it does, how do you target it? And I think that over the past several years, there's been tremendous improvement in understanding of what is Bcl-2 doing, and now we have some very specific tools in which to inhibit that protein. Now in CLL specifically, it's been peculiar, and because when you look at CLL, we do not see these translocations or alterations in the gene Bcl-2, but we see higher levels of Bcl-2 protein in CLL than we find in related lymphomas where we see these translocations.
It's overexpressed, isn't it?
It's overexpressed. And part of that reason came about with discoveries that, working with Carlo Croce in the consortium, was identified as being one of the primary genetic lesions within CLL that accounts for maybe 50 to 60% of all cases. And that is a deletion in or alterations in regulatory RNA. And one of the primary jobs of the regulatory RNA is to lower the Bcl-2 expression. And so here it is, a cancer where we've not found structural changes in the gene, but in how the gene is regulated. And that may account for why the protein is highly expressed, but I think it also underlies a very important function to this protein. So what does Bcl-2 do? Well, Bcl-2 actually we know now is a family of proteins, and this family of protein is very effective in inhibiting cell death. And it's used quite commonly in the immune system, and it's turned on and turned off, and it is turned on in cases where you want to have the cell survive all the genetic challenges of how, when the immune system goes on, there's a lot of genetic rearrangements and mutation because we have to develop an immune response to whatever is out there. And so there's a lot of genetic gymnastics that goes on. And to protect the cells from dying during that exercise, you have the upregulation of these proteins. However, when something goes awry or you don't make the right type of response or right type of antibody, then there's a need to clear the cell, and so therefore you can counteract the Bcl-2. And so this is a very important part of physiology, and the CLL cells adapted that to have a very high level of this protein. And what it does is that it makes the cell more resistant to being killed, either with chemotherapy or other drugs, and even by itself, just spontaneously. And so there's been a lot of interest in developing agents, and several years ago it was thought maybe we can deregulate the gene or downregulate it. So there was work with these small pieces of DNA, antisense DNA, and this proved to be very challenging in terms of getting the right amount into the cell and having therapy that was truly effective. Now, actually, as work has gone on, there's been a very important discovery which is now being put into clinical practice, where the Bcl-2 protein is somewhat like a lock and key. And so there are proteins of this family that are very important for triggering cell death, and the Bcl-2 is acting like a safety lock, like you would have a safety on a gun. And so by understanding that lock-key relationship, you can get a small molecule to go right into that lock and prevent it from doing its job. And so therefore, these proteins that the CLL cell has, which make it susceptible to dying, that are stimulated by part of its nature in terms of the stimulation with the immune complex, when you take off the safety switch, the cells are more sensitive than normal cells by far in terms of undergoing cell death. And so the old thinking was that, okay, we only had to inhibit Bcl-2, and then after a while we began to understand that there's a whole family of these safety switches. So the new thinking became, well, we have to inhibit the entire family of these proteins. And that was done with drugs such as ABT-263, navitoclax, and we conducted a number of studies with a number of groups, and this proved to be actually very effective. I was very impressed with the activity of the drug because I saw patients who had failed all forms of therapy. I'll never forget a woman who came down from a local community, Bakersfield, who could not even lie flat because she had so much tumor in her abdomen. And giving this orally active drug, I saw tumor shrinkage within a fortnight, and she achieved a very excellent response. Not a complete one, but I think this was encouraging. Now, what we had with the 263 was a drug that inhibited multiple members of this family, and we had problems in that that can affect other cells like platelets. And so at this meeting, they're starting to discuss, and we'll see more about this. This is a future thing to keep in track of, is the new thinking, which is that we only really have to inhibit this one protein, Bcl-2. And I think that this is actually showing some very dramatic responses in patients on clinical trials now. And so this has been a very exciting development. Now, the other developments that are, of course, very exciting are inhibitors of signaling pathways within the leukemia cell. We know it's a B cell, and a B cell is a very fine-tuned machine. It's actually very sensitive to signals that it receives from the environment, and it's a cell that's almost programmed to undergo cell death.
So you're talking about the B cell receptors. Let me ask you to hold just for a second there.